Next topic, deregulation of the cell cycle and genome maintenance pathways in cancer. The complex mechanisms that regulate the eukaryotic cell cycle are prime targets for oncogenic mutations. Both positively and negatively acting proteins precisely control the entry of cells into the cell cycle and their progression through it. In addition, cells harbor surveillance mechanisms known as checkpoint pathways that ensure that cells do not enter the next phase of the cell cycle before the previous one has been correctly completed. For example, cells that have sustained damage to their DNA are normally arrested before their DNA is replicated, or in G2 before chromosome segregation. This arrest of the cell cycle allows time for the DNA damage to be repaired. Alternatively, cells are directed to commit suicide via apoptosis. The cell cycle control and checkpoint systems function to prevent cells from becoming cancerous. As might be expected, mutations in the system often lead to abnormal movement of or contribute to cancer. In this section, we discuss the cell cycle checkpoint pathways that are affected in cancer. We first describe how the checkpoint pathway that controls entry into the cell cycle is mutated and misregulated in most human cancers. We then discuss how P53 prevents tumorigenesis by helping cells to respond to DNA damage. We end with the discussion of how defects in DNA repair enzymes contribute to cancer by compromising the cell's ability to repair DNA damage. So, mutations that promote unregulated passage from G1 to S phase are oncogenic. Once a cell progresses past a certain point of G1 called the restriction point, it becomes irreversibly committed to entering S phase and replicating its DNA. Cyclin DS, cycling dependent kinase CDKs, and the RB protein are all elements of the control system that regulates passage through the restriction point. The pathway that controls entry into the cell cycle is estimated to be misregulating in approximately 80% of human cancers. At the rate of this pathway are cycling DCDK4 by 6 complexes and the transcription inhibitor RB. The expression of cyclin D gene is induced by many extracellular growth factors or mitogens. These cyclins assemble with partner CDK4 and CDK6 to generate catalytically active cyclin CDK complexes whose kinase activity promotes progression through G1. Mitogen withdrawal prior to passage through the restriction point leads to accumulation of two CDK inhibitors, and those two proteins P15 and P16 bind to cyclin DCDK4 by 6 complexes and inhibit their activity, thereby causing G1 arrest. The transcription inhibitor RB is controlled by cyclin DCDK4 by 3 phosphorylation. Non-phosphorylated RB binds to E2F transcription factors which stimulate transcription of genes encoding proteins required for DNA synthesis. Under normal circumstances, phosphorylation of RB protein is initiated midway through G1 by active cyclin DCDK4 by 3 complexes. RB phosphorylation is completed by cyclin ECDK2 complexes in late G1, allowing release and activation of E2FS and progression from G1 to S. Inactive kinases there P16, CYCD, CDK4, P16, CYCD, CDK4, RB, E2F is there. Then activator of transcription of proteins needed for DNA synthesis. Restriction point control. Non-phosphorylated RB protein binds transcription factors collectively called E2FS and thereby prevents E2F mediated transcriptional activation of many genes whose products DNA polymerases are required for DNA synthesis. The kinase activity of cyclin DCDK4 by 6 phosphorylates RB, thereby inactivating RB and activating E2FS. Cyclin DCDK4 by 6 activity is inhibited by P16. Overproduction of cyclin D, a positive regulator, or loss of the negative regulators P16 and BR commonly occurs in human cancers. And the complete phosphorylation of RB and its dissociation from E2Fs irreversibly commits the cell to DNA synthesis. Most tumors contain an oncogene mutation that causes the overproduction or loss of one of the components of pathway that controls entry into S phase, so that the cells are propelled into S phase in the absence of the proper extracellular growth signals. For example, elevated levels of cyclin D1, one of the three cyclin Ds, is found in many human cancers. In certain tumors of antibody producing B lymphocytes, the cyclin D1 gene is translocated such that its transcription is under the control of an antibody gene enhancer, causing elevated cyclin D1 production throughout the cell cycle, irrespective of extracellular signals. This phenomenon is analogous to the MYC translocation in Burkitt's lymphoma cells discussed earlier. That cyclin D1 can function as an oncoprotein, that's shown by studies with transgenic mice in which the cyclin D1 gene was placed under the control of an enhancer specific for mammary gland cells. Initially, the duct cells underwent hyperproliferation and eventually breast tumors developed in this transgenic mice. A second mechanism that can lead to overproduction of cyclin D is gene amplification. Amplification of the cyclin D1 gene and concomitant overproduction of the cyclin D1 protein is common in human breast cancers. The extra cyclin D1 helps to drive cells to the cell cycle. We have already seen that inactivating mutations in both RB alleles lead to childhood retinoblastoma, a relatively rare type of cancer. However, loss of RB gene function is also found in more common cancers that arise later in life, carcinomas of lung, breast, and bladder. These tissues, unlike retinal tissue, probably produce other proteins, P107 and P130, both structurally related to RB, whose functions is relevant with that of RB, and this RB is not so critical for preventing cancer in these tissues. In the retina, however, regulation of cell cycle entry appears to rely exclusively on the RB protein, which is why patients heterozygous for the RB gene first develop tumors in this tissue. RB function can be eliminated not only by inactivating mutations but also by the binding of inhibitory protein designated E7 that is encoded by human papilloma virus, another nasty viral trick to create virus producing tissue. At present, this binding is known to occur only in cervical and oropharyngeal cancers. The proteins that function as cyclin CDK inhibitors play an important role in regulating the cell cycle. In particular, loss of function mutations that prevent P16 from inhibiting cyclin DCDK 4x6 kinase activity are common in several human cancers. Loss of P16 mimics overproduction of cyclin Ds. Thus, P16 normally acts as a tumor suppressor, although the P16 tumor suppressor gene is deleted in some human cancers. The P16 sequence is normal in others. In some of these later cancers, lung cancer, the P16 gene or genes encoding other functionally related proteins is inactivated by hypermethylation of its promoter region, which prevents its transcription. What promotes this change in the methylation of P16 is not known, but it prevents production of this important cell cycle control protein. The locus encoding P16 is highly unusual in that it encodes no less than 3 tumor suppressor genes, which makes it the most vulnerable locus in the human genome to oncogenic changes. In addition to harboring the P16 encoding gene INK4A CDKN2A, it has the INK4B CDKN2B locus immediately upstream, which encodes P15, another cyclin DCDK 4x6 inhibitor. In addition, the locus encodes a key activator of the tumor suppressor P53. This protein, P14ARF, P19ARF in the mouse, is encoded by an axon upstream in the first INK4A exon and shares its exon 2 and exon 3 with INK4A. As we will see next, the protein controls the stability of P53. These mutations in this locus can simultaneously affect the two major tumor suppressor pathways in the cell, the RB and P53 pathway. The protein p53 is a central player in tumorigenesis. It is thought that most, if not all, human tumors have mutations either in p53 itself or in proteins that regulate p53 activity. Cells with functional p53 becomes arrested in G1 when exposed to DNA damaging radiation, whereas cells lacking functional p53 do not. Unlike other cell cycle proteins, p53 is present at a very low level in normal cells because it is extremely unstable and rapidly degraded. Mice lacking p53 are largely viable and healthy except for the predisposition to develop multiple types of tumors. In normal mice, the amount of p53 protein is highlighted. A post-transcriptional response only in stressful situations such as exposure to UV or radiation, heat, or hypoxia. Irradiation by rays creates lesions in the DNA. Serine kinase ATM or ATR are recruited to the sites of damage and are activated. They then phosphorylate p53 on a serine residue in the end terminus of the protein. This phosphorylation causes the protein to evade ubiquitin mediated degradation, leading to marked increase in its concentration. Then arrest in G1 in response to DNA damage. The kinase activity of ATM is activated in response to DNA damage due to various stresses, UV radiation, heat. Activated ATM then triggers 3 pathways leading to arrest in G1. CHK2 is phosphorylated and in turn phosphorylates CDC25A, thereby making it for degradation and blocking its role in CDK2 activation. In a second pathway, phosphorylation of p53 stabilizes it, permitting p53 activated expression of genes encoding proteins that cause arrest in G1, promote apoptosis, or participate in DNA repair. The third pathway is another way of controlling the pool of p53. The MDM2 protein in its active form can form a complex inactive sequence in which p53 inhibiting the transcription factor and causing p53 ubiquitinization and subsequent proteasomal degradation. ATM phosphorylates MDM2 to inactivate it, causing increased stabilization of p53. In addition, MDM2 levels are controlled by a p14ARF, p19ARF in the mouse, which binds MDM2 and sequesters it in the nucleolus, where it cannot access p53. The p14ARF gene is induced by high levels of mitogenic signaling, which are frequently observed in cells carrying oncogenic mutations in growth factor signaling pathways. The human MDM2 gene is frequently amplified in sarcomas, which presumably causes excessive inactivation of p53. Similarly, p14ARF is also found mutated in some cancers. Then, deregulation of cell cycle and genome maintenance pathways in cancer. The stabilized p53 activates transcription of the gene encoding p21, which binds to and inhibits mammalian cyclin E-CDK2. As a result, cells with damaged DNA are arrested in G1, allowing time for DNA repair by the mechanisms, and are permanently arrested, that is, they become senescent. The activity of p53 is not limited to inducing cell cycle arrest. However, in addition, this multi-purpose tumor suppressor stimulates the production of pro-apoptotic proteins and DNA repair enzymes. Senescence and apoptosis may in fact be the most important means through which p53 prevents tumor growth. The activity of p53 is normally kept low by a protein called MDM2. When MDM2 is bound to p53, it inhibits the transcription activating ability of p53, and at the same time, because it has E3 ubiquitin ligase activity, catalyzes the ubiquitinization of p53, thus targeting it for proteasomal degradation. Phosphorylation of p53 by ATM or ATR displaces bound MDM2 from p53, thereby stabilizing it. Because the MDM2 gene is itself transcriptionally activated by p53, MDM2 functions in an autoregulatory feedback loop with p53, perhaps normally preventing excess p53 function. The MDM2 gene is amplified in many sarcomas and other human tumors that contain a normal p53 gene. Even though functional p53 is produced by some tumor cells, the elevated MDM2 levels reduce the p53 concentration enough to abolish the p53 induced arrest in G1 in response to irradiation. A key regulator of MDM2 is the P14ARF protein encoded by the multi-tumor suppressor locus that also encodes the INK4 proteins. The P14ARF protein binds to MDM2 and sequesters it in the nucleolus away from P53. Normal P14ARF levels are so low in tissues that the protein is barely detectable. Otherwise, it would cause P53 accumulation and hence cell cycle arrest or apoptosis. A key regulator of MDM2 is P14ARF protein encoded by the multi-tumor suppressor locus that also encodes the INK4 proteins. The P14ARF protein binds to MDM2 and sequesters it in the nucleolus away from P53. Normal P14ARF levels are so low in tissues that the protein is barely detectable. Otherwise, it would cause P53 accumulation and hence cell cycle arrest or apoptosis. However, in response to oncogenic signaling, that is, in the presence of high levels of protoproliferation signals, P14ARF transcription is induced by the E2F transcription factor. This P14ARF is an important inhibitor of tumorigenesis since it induces P53 activation when proliferation signaling reaches unphysiologically high levels through hyperactivating mutations in the signaling pathways. For proliferation signaling pathways to cause uncontrolled proliferation, as seen in cancer, this P53 upregulation must not occur. It is therefore not surprising that P53 is inactive in most human tumors through loss of P53 function itself. Downregulation of positive regulators of P53 function such as P14ARF or upregulation of negative regulators of P53 such as MDM2. The activity of P53 is also inhibited by a human papilloma virus HPV protein called E6. HPV encodes two proteins that contribute to its ability to induce stable transformation and mitosis in a variety of cultured cells. Those proteins E6 and E7 bind to and inhibit the P53 and RB tumor suppressor respectively. Acting together, E6 and E7 are sufficient to induce transformation in the absence of mutations in cell proliferation regulatory proteins. The activity of P53 is also inhibited by a human papilloma virus HPV protein called E6. HPV encodes two proteins that contribute to its ability to induce stable transformation and mitosis in a variety of cultured cells. These proteins E6 and E7 bind to and inhibit the P53 and EB tumor suppressors respectively. These proteins E6 and E7 bind to and inhibit the P53 and RB tumor suppressors respectively. Acting together, E6 and E7 are sufficient to induce transformation in the absence of mutations in cell proliferation regulatory proteins. The active form of P53 is a tetramer of 4 identical subunits. The missense point mutation in one of the two P53 alleles in a cell can abrogate almost all P53 activity because virtually all the oligomers will contain at least one defective subunit, and such oligomers have reduced ability to activate transcription. Oncogenic P53 mutations thus act in a dominant negative manner in which a single mutant allele causes a loss of function. The loss of function is incomplete, so in order to grow more rapidly, tumor cells still sometimes lose the remaining functional allele, loss of heterozygosity. As we learned in chapter 6, dominant negative mutations can occur in proteins whose active forms are multimeric or whose function depends on interactions with other proteins. In contrast, loss of function mutations in other tumor suppressor genes are recessive because the encoded proteins function as monomers and mutation of a single allele has little functional consequence. The P53 protein is a key defense mechanism against oncogenic transformation. This is best illustrated by the observation that loss of function mutations in the P53 gene occur in more than 50% of human cancers. What does P53 protect us against? Unlike RB, which prevents inappropriate proliferation, P53 guards the cell from genetic changes. When the P53 G1 checkpoint control does not operate naturally, damaged DNA can replicate, generating mutations and DNA rearrangements that are passed on to daughter cells and make their transformation into metastatic cells more likely. What does P53 protect us against? Unlike RP, which prevents inappropriate proliferation, P53 guards the cell from genetic changes. When the P53 G1 checkpoint control does not operate properly, damaged DNA can replicate, generating mutations and DNA rearrangements that are passed on to daughter cells and make their transformation into metastatic cells more likely. For example, loss of P53 function leads to a 100-fold or greater increase in the frequency of gene amplification. At the same time, loss of P53 function prevents apoptosis, contributing to evolution of transformed cells. Because of its central role in preventing tumorigenesis, researchers are intensely searching for compounds that can restore P53 function as a new way of treating a broad spectrum of human tumors. Then loss of DNA repair systems can lead to cancer. As our previous discussion has shown, alterations in DNA that lead to the malfunction of tumor suppressor proteins and the production of oncoproteins are the underlying cause of most cancers. These oncogenic mutations in key growth and cell cycle regulatory genes include insertions, deletions, and base substitutions, as well as chromosomal amplifications and translocations. Damaged DNA repair systems lead to an increased rate of these genetic alterations. Of the many mutations that accumulate in cells with defects in DNA repair mechanisms, some affect cell cycle regulators, some cell adhesion, and some the ability to migrate through basement membranes. Discussed in the beginning of this chapter, cells that have accumulated these kinds of mutations may become cancerous. Furthermore, some DNA repair mechanisms themselves are error prone. Those errors also contribute to oncogenesis. The inability of tumor cells to maintain genomic integrity leads to the formation of a heterogeneous population of malignant cells. For this reason, chemotherapy directed toward a single gene or even a group of genes is likely to be ineffective in wiping out all malignant cells. This problem aids to interest in therapies that interfere with the blood supply of tumors, target aneuploid cells, or in other ways act on multiple types of tumor cells. Normal dividing cells usually employ several mechanisms to prevent the accumulation of detrimental mutations that could lead to cancer. One form of protection against mutations from stem cells is their relatively low rate of division, which reduces the possibility of DNA damage incurred during DNA replication and mitosis. Furthermore, the progeny of stem cells do not have the ability to divide indefinitely. After several rounds of division, they exit the cell cycle, reducing the possibility of mutation-induced misregulation of cell division associated with dangerous tumors. Furthermore, if multiple mutations are required for a tumor to grow, attract a blood supply, invade neighboring tissues, and metastasize, a lower rate of replication combined with the normal low rate of mutations provides further shielding from cancer. However, the safeguards can be overcome if a powerful mutagen reaches the cells, or if DNA repair is compromised and mutation rate rises. When cells with stem-like growth properties are mutated by environmental poisons and are unable to effectively repair the damage, cancer can occur. Even without exposure to any external carcinogens or mutagens, normal biological processes generate a large amount of DNA damage. That damage is due to depurination reactions or alkylation reactions and to the generation of reactive species such as oxygen radicals, all of which alter DNA. It has been estimated that in every cell, more than 20,000 alterations to the DNA occur each day from reactive oxygen species and depurination. This DNA repair is a crucial defense system against genetic change and hence against cancer. The normal role of genome maintenance genes is to prevent or repair DNA damage. Loss of high fidelity DNA repair systems that are described in Chapter 5 correlates with increased risk for cancer. For example, people who inherit mutations in genes that encode a crucial mismatch pair or excision repair protein have an enormously increased probability of developing certain cancers. Without proper DNA repair, people with xeroderma pigmentosum or hereditary non-polyposis colorectal cancer, also known as Lynch syndrome, have a propensity to accumulate mutations in many other genes, including those that are critical in controlling cell growth and proliferation. XPCOS affected people to develop skin cancer at about 1,000 times the normal rate. 7 of the 8 known XP genes encode components of the excision repair machinery, and in the absence of these repair mechanisms, genes that control the cell cycle or otherwise regulate cell growth and death become mutated. HNPCC genes encode components of mismatched repair system, and mutations in these genes are found in 20% of sporadic colon cancers. The cancers progress from benign polyps to full-fledged tumors much more rapidly than usual, presumably because the initial cancer cells are undergoing continuous mismatch mutations without repair. One gene frequently mutated in colon cancer because of the absence of mismatch pair encodes the type 2 receptor for TGF beta. The genes encoding this receptor contains a sequence of 10 adenines in a row. Because of the slippage of DNA polymerase during replication, the sequence often undergoes mutation to a sequence containing 9 or 11 adenines. If the mutation is not fixed by the mismatched repair system, the resultant frame shift in the protein coding sequence abolishes production of the normal receptor protein. As noted earlier, such inactivation mutations make cells resistant to growth inhibition by TGF beta, thereby contributing to the unregulated growth characteristics of these tumors. This finding arrests the importance of mismatch repair in correcting genetic damage that might otherwise lead to uncontrolled cell proliferation. All DNA repair mechanisms use a family of DNA polymerases different from shared pol alpha, pol delta, and pol replicative DNA polymerases to correct DNA damage. None of these polymerases, including one called DNA polymerase beta, are capable of using templates that contain DNA adducts and other chemical modifications, even missing bases. These enzymes are called Lasian bypass DNA polymerases. Each number of this polymerase family has distinct capabilities to cope with particular type of DNA lesions. Presumably, such polymerases are tolerated because often any repair is better than none. They are the polymerase of last resort and ones used when more conventional and accurate polymerases are unable to perform, and they carry out a mutagenic replication process. DNA pol beta does not proofread and is overexpressed in certain tumors, perhaps because it is needed at high levels for cell to be able to divide at all in the face of a growing burden of mutations. Error-prone repair systems are thought to mediate much, if not all, of the carcinogenic effect of chemicals and radiation, since it is only after the repair that a heritable mutation exists. There is growing evidence that mutations in DNA pol beta is associated with tumors. When 189 tumors were examined, 58 had mutations in the DNA pol beta gene, and most of the mutations were found neither in normal tissue from the same patient or from the normal spectrum of mutations found in different people. Expressing of two mutant polymerase forms in mouse cells caused them to grow with a transformed appearance and an ability to form foci. Double-stranded breaks are particularly severe lesions because concurrent rejoining of double-stranded DNA can lead to gross chromosomal arrangements. Rearrangements and translocations, such as those that produce a hybrid gene or bring a growth regulatory gene under the control of a different promoter or enhancer. Often the repair of such damage depends on using the homologous chromosomes as a guide. The B and T cells of the immune system are particularly susceptible to DNA rearrangements caused by double-stranded breaks created during rearrangement of their immunoglobulin or T cell receptor gene, which explains the frequent involvement of this loci in leukemias and lymphomas. BRCA1 and BRCA2 genes implicated in human breast and ovarian cancers encode important components of DNA break repair systems. Cells lacking either of the BRCA functions are unable to repair DNA, where the homologous chromosome is providing the template for repair. Then key concepts of this section: deregulation of cell cycle and genome maintenance pathways in cancer. Overexpression of the proto-oncogene encoding cyclin D1 or loss of the tumor suppressor gene encoding P16 and RB can cause inappropriate unregulated passage through the restriction point. Such abnormalities are seen in 80% of human tumors. The INK4ARF locus represents the most frequently mutated tumor suppressor locus in humans, controlling both the RB and P53 pathways. P53 protein is a multifunctional tumor suppressor that promotes arrests in G1 and DNA repair or apoptosis in response to damaged DNA. Loss of function mutations in the P53 gene occur in more than 50% of human cancers. overproduction of MDM2, a protein that normally inhibits the activity of p53, or inactivation of p14ARF, which also increases MDM2 activity, occur in several cancers that express normal p53 protein. This, in one way or another, the p53 stress response pathway is inactivated to allow tumor growth. Then human papillomavirus encode two carcinogenic proteins, E6 which inhibit p53 and E7 which inhibits RB. Genome maintenance genes encode enzymes that repair DNA or otherwise maintain the integrity of chromosomes. When DNA damage does occur, mutations in genome maintenance genes lead to a high rate of mutagenesis of the genome that can lead to uncontrolled cell proliferation and accumulation of additional mutations, resulting in progression to metastatic cancer. Inherited defects in DNA repair processes found in certain human diseases are associated with an increased susceptibility for certain cancers. So that's all about this topic of the cancer. So thank you for listening.